Uh, 2020 possibly in the human history. It is an unprecedented COVID-19 pandemic year. So our life, activity, everything uh, changed uh, dramatically, and a new normal now we are experiencing, and day by day, maybe you know, many online activity would be normal. So I welcome both of you to our course BTL 611 probiotics, prebiotics, and postbiotics. This is, in fact, a new course at uh, BSMRAU. Possibly, you cannot find this a similar title of the course anywhere in Bangladesh. And I am happy that at least two students, one from uh, masters, another PhD, are now attending. Uh, in our previous lecture, we discussed about plant probiotics. Yesterday, I went through some of the literature, plant probiotics as well as microbiome. They are now a, an interesting subject for advanced research. So I will share two papers, one published in Nature Biotechnology and another paper published in Nature Microbiology Reviews. And some of the highlights I will show where they focused the probiotic microorganism. One paper, possibly you have noticed that it was published possibly in Science or Nature, that in America, some obese people, fecal samples from the obese people transferred to the transplant to the from the healthy people transplant to the obese people for several times, and obese people person gradually become normal. That means microbiota or microbiome in our intestine as well as in the plant. They are crucial for the uh, well-being, that is a good health as well as fitness of the host plant or other eukaryotic organisms, animals. Uh, Very crucial. This is why I think it could be one of the powerful area for our research, because it is an emerging area of research. And fortunately, in our country and our university, Dr. Nazmul, he recently graduated from University of Dhaka. I was his examiner, and he did a fantastic job in whole metagenomic analysis of mastitis infected cows, and he knows how to handle whole metagenome. Metagenome means perhaps you know this term. Metagenome means most of the microorganism we cannot cultivate in the laboratory. Um, we can cultivate maximum 5% of the microorganisms available in this earth in different ecosystems. This is why scientists developed a new method, a next generation sequencing technology, where you can extract the DNA of any sample, soil or plant or human fecal samples or anything else, for milk sample, and extract DNA of all organisms. And then you can sequence all the DNA by using next generation sequencer. And fortunately, we have such a powerful sequencer in our lab. After sequencing, you will get the mixture of DNA sequence of various organisms, maybe hundreds of organisms. Uh, and bioinformatics software can discriminate, you can make group for example, bacteria, fungi, nematode, virus, and others. And among the bacteria, then you can again analyze to identify them in different groups, like different genus. Even you can go up to the species level. And this is really an amazing technique. Even unculturable microorganisms, we can now get their sequence data. And from the sequence data, we can get their proteomics data or enzyme they produce or metabolites they produce because, genome contains different genes and gene express through uh, transcribing into the mRNA. mRNA then synthesize protein. Some of the proteins are enzymes. They involve in different metabolic pathway and generates different metabolites. So any gene expression is a multi-step pathway. If you get any trace of proteome, then you can go back to the gene responsible for that proteome. Uh, they are interconnected because specific code recruit the specific amino acid in the protein, and during gene expression the mRNA contains secret codes, three-letter codes for recruiting each amino acid and getting from the gene. uh, this is why we can now very powerful by using different bioinformatics softwares to understand the mystery of life. This is why I am thinking to organize some day-long workshop to train you guys some more, maybe around 10 people, uh, by Dr. Nazmul and some other expert in bioinformatics, so that you can understand how to handle the data of uh, you know uh, biological data, genomics or other omics data, transcriptomics or metabolomics data. That would be fantastic because this era is, the era of genomics, phenomics, as well as all omics technology. Even in a given time, the ions present in the any cell is called ionomics, like nitrogen, phosphorus, potash. So omics technology is now the most popular and powerful technology, and they are interconnected and interrelated by using software, computer software. We can uh, uh, establish the relationship between them and understand the molecular mechanism of the phenotype or phenotypic changes that we can see. Let me show you some of the slides. Uh, in the recent past, one of the devastating disease we experienced in Bangladesh is called wheat blast, and it is a clear threat to food security of Bangladesh as well as South Asia, because South Asian country, our neighbor India, is the second largest wheat producing country in the world. China is number one wheat producing country. Pakistan also produce significant amount of wheat. Wheat is really a very, very important crop in the world, which offers 20% calorie among the total calorie consumed by the human all over the world, and 25% protein from the cereals. When here you can see in the field, all the spikes became white, and that means they were free from any grain, no grain formation. But plant looks like the bottom lower part you can see green, and 100% of the spikes are infected. It was an amazing synchronous uh, natural dissemination of the pathogen. 100%, even if we do artificially by using conidia of fungus, it is impossible. But in nature, wheat blast is like that, and it has been in South America since 1985. But in 2016, it landed in Bangladesh, and Bangladesh now, due to wheat blast, it is fifth highest wheat importing country, and our wheat production dramatically decreased 5 lakh tons. And wheat blast first appeared in 8 districts of Bangladesh. Later, it spread into 20 districts out of, first they were here. And you see, when seeds are infected plant seeds are not edible, even not edible by the animals or like that. Yield loss was 100%. And the wheat blast fungus produce different toxin that kill the plant. And here is the conidia of the wheat blast fungus. So wheat blast fungus uh, in our laboratory is very famous because by using field pathogenomics and international collaboration, we first determined the origin of the wheat blast by using field pathogenomics approaches. And we confirm that our wheat blast strains are clonal, all are clonal, same, isolate genetically. Uh, and they came from South America. That is a Brazilian isolate like PY 0925, very close to our isolates. And this discovery helped us to take management option, because as we diagnosed it properly, then we understood that what kind of measures we have to be taken. And South America, they have been working on wheat blast since 1985. So we readily use their different uh, agronomic and other cultural practices to manage the disease. This is why after 2016, the disease was not so severe, and weather also was not so favorable, because wheat blast requires rain or heavy dew fall so that plant can be wet for at least 20 hours for the epidemic outbreak. And I mentioned that we published a paper in BMC Biology, and that was, cited 196 times meanwhile. And we targeted how can we control the wheat blast, whether we can use our probiotic bacteria, because we have a big stock of probiotic bacteria, and we tested them against the wheat blast fungus. And we found that four isolates displayed strong antagonistic effects toward wheat blast fungus, Megnaporte origin triticum. So this is our story, our work. I shall show you later on some of the more details of our for application of our four isolates in controlling wheat blast. But now I would like to share some of the basic research report published in Nature Biotechnology. Here you see rhizosphere microbiome structure alters to enable wilt resistance in tomato. In tomato, Ralstonia solanaceae is a dangerous soil-borne pathogen, and they found that tomato variety Hawaii 7996 is resistant to Ralstonia solanaceae, but moneymaker variety is susceptible. So another variety is susceptible. So to evaluate whether plant-associated microorganisms have a role in disease resistance, we analyze the rhizosphere microbiomes of both variety in a mesocosm experiment. Microbiome structure differed between the two cultivars. transplanting of rhizosphere microbiota, you see, from the resistant one to the susceptible one. They found suppressed disease symptoms in susceptible plants. Comparative analysis of rhizosphere metagenomes from resistant and susceptible plants enabled the identification and assembly of a uh, flavobacterial genome that was far more abundant in the resistant plant rhizosphere microbiome than in that of the susceptible plant. We cultivated this flavobacterium named TRM1 and found that it could suppress Ralstonia solanacearum disease development in a susceptible plant in pot experiment. Our findings reveal a role for native microbiota in protecting plants from microbial pathogens, and our approach charts a path forward toward the development of probiotics to ameliorate plant disease. You see, this is a simple job. Actually, I thought this type of job since long to do in our lab, but we had no, uh, uh, next generation sequencers and other facilities. We have now, wheat blast resistant variety, 100% resistant variety. We have now moderately resistant variety. We have now susceptible variety. Variety means genotypes. 100% resistant, it is not a variety, it is a genotype we received from Japan. And it contains RMG GR119 and gene. People thought it is responsible for the a resistance against the wheat blast. But here, this result telling me something different. Maybe microbiome present in the RMGGR119 genotype, they may help to express that resistant gene, or somehow they also contribute to their resistance. So it is very important, not only looking a narrow view that is only gene is responsible for the resistance, like Hawaii 7996 resistant variety. They found completely different microbiome than the susceptible variety of tomato moneymaker. So in our case, it is a nice subject to do whole microbiome in resistant variety, susceptible variety, and moderately resistant variety. And I'm expecting uh, that we can find differential microbiome. And if we can prove that after isolating the target microbiome in the laboratory, that microbiome also plays a role in suppressing the wheat blast disease. And if we can do the research after inoculation of the bacterium, which is really a dominant player in the resistance. And if we can show that it is associated with the expression of the gene in the RMGGR119 variety, then it could be published in Nature, because we can then tell that microbiome uh, present in the resistant plant also trigger gene expression in the host against the pathogen. Not only they use different chemical weapons. Okay, here you can see they analyze the comparison of the soil community structure. They analyze the microbiome in Hawaii 7996 variety and moneymaker variety, as well as Korean cabbage variety, a control, and also in the bulk soil. Bulk soil means free from the soil where plants are not cultivated. Here you can see there are huge difference in proteobacteria, as well as different bacterio that is and other bacteria. And very interestingly, the difference is very unique. And based on the differences, you can imagine that which players are linked to the resistance and which are dominating in the resistant variety. And they analyze the population in different varieties and try to make a correlation of the disease. Here you can see before transplanting. That means Hawaii 7996 variety. This is resistant, no disease, Ralstonia, Solanacerium bacterial disease. But moneymaker, you see, it is disease. The color difference showed the disease. It is a illustration, just a diagram. And then when this is same thing. But what happened, you see, when they transplant rhizosphere soil from Hawaii 7996 to the moneymaker here. Here you see it is how to say resistant. And when transplant moneymaker contains microbiome differently to the Hawaii, it showed the susceptible. That means this data uh, is telling that genetic makeup of the plant is not only responsible for their resistance. Microbiome, associated microbiome is important. You can ask me a question, why microbiome is different? Different because it is associated with the genetic makeup of the plant also, because genetic makeup is responsible for release of carbon containing compound, because plant have the ability to do photosynthesis and then produce lots of metabolites in their carbon containing compound in their cell from the photosynthetic, like the light energy converted into the chemical energy. And nearly 40% of the photosynthetic carbon containing compound they release through the root to offer the carbon source, that is energy source, to the microbiome. So genetically these two plants different. This is why they are excreted root exudate may contain different metabolites, and different metabolites are good for different microorganisms. This is why diversity is there. And this is really an amazing research, and they nicely depicted it. In case of our, we can design an experiment in wheat like this way. So plant microbiome interactions from community assembly to plant health. This is a review article published in Nature, review in microbiology. What they say, healthy plants host diverse but taxonomically structured communities of microorganisms, the plant microbiota. That colonize every susceptible plant tissue, accessible plant tissues. That not only the root, but every accessible plant tissues. Plant-associated microbiome confer fitness advantages to the plant host, including growth promotion, nutrient uptake, stress tolerance, resistance to pathogen. In this review, we explore how plant microbiome research has unraveled the complex network of genetic, biochemical, physical, and metabolic interactions among the plant, the associated microbial communities, and the environment. That means tripartite interactions are taking place. One is plant, another is microbial communities, another is environment, because environment where a plant is grown creates serious stress to the plant. So microbial community as well as plant's own genetic makeup, they jointly work together to fight against the environmental stress. So stress not only pathogenic stress. Stress may be the abiotic stress like heat stress, or maybe salinity, or drought and others. We also discuss how those interactions shape the assembly of plant associated microbiomes and modulate their beneficial traits. such as nutrient acquisition, because plant cannot take up nutrient or uptake nutrient from the soil or environment without the help of microorganisms I discussed earlier, because plant requires 16 to 70 essential nutrient elements, and all of them they are uptaking in an ionic form, and microorganisms play a vital role in transforming the organic and inorganic insoluble compounds to make ionic form of nutrients. And plant health, that means disease resistance, obviously exerted by the microbiome that developed in the plant tissues. In addition to highlighting knowledge gaps and future direction, this paper I will share with you, review paper, give you a holistic understanding of the subject matter. Here you can see, in this is a maize plant. You know this plant. In maize plant, there are people analyze the above the ground part as well as the root part. And they showed here uh, above the ground and in the root, bacteria and fungi are the predominant microbiome. In the above ground, you see mostly basidiomycetes are dominating in the maize plant, and ascomycetes and other very small amount of other fungi are present. But in case of bacteria, proteobacteria are the dominant, nearly 50%. And then in different organ, you see leaf and then stem, they analyze root base or like this, and they found there are some shifting and the differences. And in the bulk soil, composition is like that. But in the rhizosphere, you see, uh, again, basidiomycetes and ascomycetes are dominant and non-pathogenic fungi. But here also, proteobacteria are dominating, but here firmicutes are also higher in the rhizosphere. And very interestingly, they found that these microorganisms play a vital role in the growth, development, fitness, as well as health of the host plant. And here, general structure of the bacterial and fungal communities, they, analyzed statistically by using this box plot method and the chart, the same thing. And this is another image, plant colonization and microbiome assembly. This is, for example, a plant root. Okay, plant root. And a plant root, exude lots of carbon containing compounds. I told you nearly 40% of the photosynthesis they exude from the root. Why? To promote the growth of the microbiome in the rhizosphere, because they are very critical for the nutrient uptake of the plant. They mineralize the inorganic as well as organic compounds, and finally offer the plant nutrient in available form. Here you see root exudates contain lots of diversified chemical compounds. Depending on the chemical structure of the composition of the root exudates, the microbial communities are established, because some of the root exudates contain different flavonoidal or signaling compound. They attract the bacteria and other microorganisms through chemotaxis and cell motility. That means attract the microorganisms as well as trigger their gene expression and their reproduction, and make a biofilm on the surface. Biofilm. You see, if a biofilm on the root surface like this, a beneficial bacterium that can produce secondary metabolites inhibitory to the pathogen, then root would be fully protected. Even this, if they are beneficial and colonized like this way, pathogenic microbes cannot get space and nutrient to colonize on the root. So here, different type of interactions taken place. Intermicrobial interactions also taken place. Microbes interaction. One, they have the competition. They release the toxin to stop others, and defense system, antibiotic resistant gene. Some of them contain CRISPR, and they have the quorum sensing system. Quorum sensing means bacteria can release small molecules called acyl homoserine lactone to talk with each other. Each other means their own community members, whether if there is enough food and environment is good, then they decide for cell division very quickly. And quorum sensing signal tells the community members all the information, even the warning of the some stressful environment. And metabolism and transport degradation, they can degrade the toxic molecules like arsenic and other heavy metal. Substrate utilization, I told you, organic and inorganic substrate that contains nutrient elements not only required for plant, for them also essential nutrient elements. They break down and make them ionic form, and they also uptake in ionic form, and meanwhile plants also uptake them. Amino acid metabolism, and endophytic colonization. Some of them can enter into the internal tissue of the plant and then colonize, inter cellular spaces, and they do not harm the plant. No symptom develops, but protect the plant. They offer the phytohormones and other important enzymes, even transcription regulators. They release some compounds that help plants to express the gene good for them. And some of them release effectors, some protein that are good for the plant defense or plant growth. So lots of molecular crosstalks are taken uh, uh, among the microorganisms in the rhizosphere, as well as microorganisms and the plants. So you can easily understood that microbiome and host plant interactions is really a very complex, system, and our understanding is still limited. But what we understood that microbiomes plays a good role, and important role, crucial role for the fitness of the plant, growth of the plant, as well as better health of the plant. Here you can see microbiome, how some of them can fix nitrogen, and those some of them can even colonized on the root tissue, like in case of legume rhizobium node, symbiosis. In that case, rhizobium bacterium, for rhizobium bacterium, plant make a nodule like this structure, house. First curve this one, and they have the crosstalk because plant release luteolin, a flavone signal to attract the rhizobium bacterium, like bradyrhizobium, and then bradyrhizobium secrete a polysaccharide to trigger the root to form the this type of nodule structure, and inside the nodule structure bacteria present, and those bacteria can easily fix atmospheric nitrogen and offer the nitrogen nutrition to the plant. But there are lots of other free living bacteria. They can also fix atmospheric nitrogen and offer to the plant. And some of the bacteria can, I told you, make insoluble nutrient into soluble form, like sulfate, ferrous, or inorganic phosphate ion, potassium ion, magnesium ion, calcium ion. They solubilize the nutrient and offer to the plant. And here you see there are green arrows coming. That means Plant also produce lots of photosynthesis, and they release through the root to the microorganism living in the rhizosphere. So bacteria or microbiomes produce stimulus, plant hormones, SEC, deaminase, enzyme, osmoprotectants, and detoxification of enzymes, lots of things they do for the stress tolerance of the plant, protect signal against the pathogen and invertebrate pest. How? Direct mechanism or indirect? Direct secretion system, secretion of different antibiotics, or they have the pili. Pili means fine protein appendages on the surface of the microorganism like bacteria, so they directly fight against the pathogenic microorganisms. Indirectly, competition for the space, they produce siderophores. Siderophores are secondary metabolites. They attract the iron and for plant uptake, and pathogenic microorganism suffers from iron and they cannot grow. Lytic enzymes they secrete, and lytic enzyme can cause lysis of the pathogen cell. Antibiotic secretion and volatile compounds they also secrete to stop the pathogen. And indirectly, they also induce systemic resistance. Systemic resistance, that means trigger the expression of genes responsible for the resistance against the stresses. So, and it is systemic all over the body. Plant can show this type of activities. Plant dysbiosis, microorganism and microorganism interactions, that means pathogenic interactions also triggered by the plant interaction, and as a result plant growth and mitigation of abiotic stress, defense against the pathogen, everything can be possible. Now the question is, you may ask question, why agriculturists are not exploiting all these phenomena as new technologies? Because our understanding in plant microbe interactions has started to uh, uh, begun in the light, because it was a dark world, and metagenomic analysis, next generation sequencing, and different bioinformatic analysis giving us to shed light on these very complex interactions, and to exploit all these beneficial traits of the interactions between and among the microorganisms, between the microorganisms and the plants. We need more knowledge. This is why I can tell you this subject is an emerging subject, and in future you can see even engineering will be done to promote the colonization of the beneficial bacteria in the host, not only direct insertion of the resistant genes or like that. So these are the now new thought synthetic biology people are thinking, and hopefully in future lots of things can be done. So in our case, we need to do some of the research because we have the good facilities. So I shall discuss later on how we are handling and using probiotic microorganisms for enhancing the plant protection against the deadly pathogens. So this is all for today in this new year, and you can ask me some question.
So it's a really interesting thing, uh, and also uh, good to know this new especially for the new publication, uh, and and it encouraged me also to do something on it.
Thank you. Actually, in case of seed industry, they are offering, good variety seed to the farmers for better yield. Nowadays, what people are doing during flowering, seed also contains lots of microorganisms. But during flowering, if you spray a good composition of bacteria, and you can change the microbiome in the seeds, and those microbiome, if they are rich in probiotic microorganisms, then those seeds will give you, vigorous plant. And during germination, all the bacteria, they will play a role, and they colonize and they dominate in any environment. And this is becoming an industrial topic soon. And the hypothesis and practical testing in the greenhouse as well as field have been done, but not yet a commercial product available. That means microbiome enriched seed. Such a business is, I think, a new business in future.
We are also facing some problems, especially for the seed germination. I would like to talk about what's called papaya and rose. They are really germination percentage is very low. So we can also introduce such type of microbiome to enhance the germination.
This is great idea. Yes, we found in case of rice and wheat and cucumber, if we add some bacteria, we can enhance the germination percentage as well as seedling vigor. We have lots of information and thesis. But in case of papaya and other, plants, we have not tried yet, but it is possible to enhance the germination as well as the growth of the seedling at initial stage by modulating the microbiome around and inside the seed. It is possible. Tomorrow I have a meeting with Bangladesh Academy of Science at around 11 o'clock. So if I take your class afternoon, is it okay? Because you have two class tomorrow. Tomorrow is Monday. So morning class, please suggest me how can I take afternoon. Anytime is okay for me. But morning I shall, heavy negotiation with Bangladesh Academy of Science for budget rationalization of a commission project.
Uh, Sorry, in your natural biotechnology paper, I've seen that the obese people and the healthy people use transformation of bacteria from healthy people to obese people. So, what are the source of bacteria in that cases? Both people are growing in the same environment. In case of human, but what will be the source?
Source
How,
means what they collected the fecal sample, from the intestine, partially digested material they took from the healthy people and transferred to the obese people, like, in case of tomato plant, rhizosphere soil transfer from one to another. In a science or nature paper, you can check and browse. I was wondered to see that how microbiome can do that, because intestinal microflora, most of them we cannot cultivate in the artificial media. They are mostly unculturable. But what they did, they transferred the partially digested food materials from the one intestine to another, that is from healthy to obese. And within 7 days, they found the effect, dramatic effect, and that was really very, a stunning phenomena. And this is why they published the paper, and they checked blood parameters and other immunity and all the things, and they found a very nice correlation.
I'm actually invested on that factor that in the within the intestine, where this bacteria comes from, whether this from parents or whether this from environment. You have mentioned in case of soil, we have lots of bacteria.
It is, I understood your point, the source of the good bacteria in the healthy person. Uh, the answer is not straightforward. There might be some genetic uh, uh, relationship also, bacteria and the host. But food habit, lifestyle, are very, very crucial. Uh, uh, Food habit and lifestyle is really, for example, Japanese people, their food habit, if you consider any person, incredibly diverse. Even in a lunch box, you can find 10 to 15 items, different types of items. But in our lunch box, you can find chicken, mutton or and rice only, like that. So food habit is very important. And genetic makeup, all people are not genetically same. So genetic makeup of a person, which inherited from generation to generation through good habit, good environment. They harbor the good bacteria and who evolved the bacteria and their host. So this also factor. There are lots of different factors are involved. Some people, who are alcoholic drinker, regularly drink too much alcohol. You can see their microflora, intestinal microflora, even microbiome quite different than non-alcoholic. Smoker microbiome should be different from the non-smoker. So it could be a good study even in the same family member. If you check the microbiome of a smoker and non-smoker, microbiome of a alcoholic drinker and non-alcoholic person, and if you can show what are the bacteria they are sensitive to those stresses, would be very nice.
I have another question. In case of wildlife or wild plant, they are surviving from thousands of years within the same condition. Whether some plants or uh, animal species have extinct, so I think there may be some interaction with the commensal bacteria, or whether this help to survive. What is your opinion in that cases?
Yes, this is a good idea because in the same species, same genus, maybe, or even same species, wild relatives, same genus you can find wild relatives, and the which is domesticated. If you compare their microbiome, you can find some good relation that why wild relatives they have the high fitness to the environmental stresses. For example, weed, undesirable plants in the field or in the bush or roadside, we are not offering any care or any nutrition, but they are growing very vigorously. And wild animals also, we never care and never bring them to the hospital, or we never offer them nutritious food as we thought, so-called nutritious food, but they are growing naturally very strong and even in the a stressful environment. Obviously, their microbiome is different. For example, if you check the microbiome of Royal Bengal tiger in Sundarban, if you can collect the sample from their very difficult job, and if you collect sample from the Royal Bengal tiger in the case or Chiriakhana, you can find obviously difference. Surely.
Thank you, sir.
Any more question?
No, sir.
Any more question?
No, sir.
If no more question, please let me know. Tomorrow's afternoon class schedule. Thank you so much.